Hi all, Dr. Clark here. Today for general biology, we're going to start talking about the inner workings or the structure of DNA. Okay, so the next few lectures is really about how DNA copies itself. What's the process by which DNA goes through DNA synthesis? So how does it unzip? How do new strands form? And the inner workings, what happens if there's a mistake, these kind of things. Before we get into that, we're going to talk about really the structure of DNA, how we know what the shape of DNA is, and for that matter, how did we discover some of those pieces. Okay? So the structure of DNA, there are actually three forms of DNA. I'm showing you two. Okay? There's the A DNA, or the A form, the B form, and then there's another form which we call the Z form. And really, it doesn't change much. DNA is double helix, okay, in all the forms. It just happens to be that in some of the forms, that double helix pattern is kinked a little bit. It's kind of at an angle a little bit. So the A form is kind of angled. It's angled, I think, at like a -1.5 degrees or so. The B form is a natural kind of straight double helix, and the Z form takes a much more angled variety. These are X-ray crystallography films, okay? And in this case, this is what made Rosalind Franklin. If you've never heard of Rosalind Franklin, she's a very famous scientist if you know much about DNA. This is what made her famous, is she was very good at taking films or taking pictures of DNA. She actually perfected it. She was probably the first person to do it and do it well. So we'll talk a little more about Rosalind Franklin and her advances, but nonetheless, these are just two forms of DNA. Alright, so before we get into the actual structure of DNA, we're going to talk about the research and kind of the methods at which researchers went about to figure out that chromosomes are carrying DNA. So remember, we were moving from Mendelian genetics, and Mendel had no knowledge of what chromosomes were or DNA or anything like that. He figured it out mathematically. Since then, we've known that there's lots of exceptions to Mendelian genetics, tons of them. But nonetheless, Morgan's work with how chromosomes segregate and crossing over, and Sutton's work on sexual chromosomes and chromosome numbers, and together those guys' work really kind of paved the way. But there was still this piece missing. And the piece missing was, we know that DNA has this connection to proteins, or at least we thought there was this connection between proteins and DNA. But we didn't know, at least in the early 1900s, we didn't know which one was the material of inheritance. Are you inheriting the proteins? Or are you inheriting the DNA? So what is genes? What are genes made of? So that's kind of one of the big questions of the early 1900s, was, can we devise an experiment to figure out, is DNA the material of genes, or are proteins the material of genes? And Frederick Griffiths in 1928 devised an experiment that kind of tested this. Okay? Not completely directly, but at least led to further experiments to support that genes are made of DNA. We now know this, okay? But at the time we didn't. Okay? So what Griffiths did was, he took a pathogenic or disease-causing bacteria, okay, Streptococcus pneumoniae, and we knew that there was two different strands of it. One strand that we say is virulent, which means it's disease-causing and will often cause death, and then one that is non-virulent. And this is very common in bacteria. So bacteria, a lot of times we'll talk about, different species of bacteria and harmful bacteria and not harmful bacteria, and it can be as simple as just a tiny little tweak in a protein, and that can go from being non-virulent to virulent. So the virulent strand he called the S form, and that is coated with a polysaccharide cap. And so we've talked about this before. Remember, bacteria will have 2 or 3 layers. First layer, plasma membrane, then a cell wall, and then potentially a capsule or a cap that goes around it. So in the case of the S form, the cap is present, and if you were to inject it into something like a mouse, it would die from that variety of Streptococcus pneumoniae. The other form is a mutated form, which we call the R form, and there is no capsule, and therefore it's non-virulent. And the individual, well, in this case a mouse, his immune system could reject this, or really it's not going to have much of an effect on the mouse. So his experiment was to do something very simple like this. You take a bunch of mice and you inject some with the live S bacteria, see what happens to them. They all die. You inject some with the live R bacteria, see what happens to them. They all live. You inject some with a heat-killed S bacteria, so you heat this up past its thermal maximum and destroy the bacteria and inject it, and the mouse lives. But then you can heat the S form, the virulent form, and combine it in solution with the R form, and the mouse dies. So what Griffiths figured out was that bacteria could transfer material from one bacteria to the next. But we know if we heat something that we're denaturing the protein. So this kind of led him to believe that it's not protein being passed from one bacteria to the other, but it's something else. And in this case, it led us to the discovery that it was DNA and not proteins. Well, that really brought another experiment to the forefront, and that was what we call the HersheyChase experiment. And Hershey and Chase were two kind of molecular biochemists that were interested in discovering, well, what is the genetic material? And they did this kind of a unique way, but it's not unique anymore. We do it all the time now, but they're kind of the first group to really look at this. And what they did was tag DNA and proteins. And what I mean by tag is you use a radioactive isotope that's found as part of DNA or that protein. So in the case of DNA, we know that DNA contains phosphorus. So what you can do is you can grow DNA in a radioactive phosphorus media, so that when the DNA is constructed and it replicates, that it picks up some of this heavy radioactive phosphorus. And then in another solution or another media, you can grow a virus which is going to contain protein, that instead of this protein having phosphorus in it, it would have a radioactive sulfur. And the sulfur is probably a binding component to the protein, helps bind it up, or a major component of the protein, and so you get this heavy sulfur molecule. By doing that, you now have in solution, you have an idea of, well, is the virus passing on DNA, so it's labeled as a different color, or is the virus passing on protein, which is labeled as another color? And that's exactly what the Hershey-Chase experiment did.
In the Hershey-Chase experiment, bacterial viruses, called phage, were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing p32 labeled deoxyribonucleotides. This resulted in a phage population with p32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the p32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
Okay, so there you can see the Hershey-Chase experiment. To us, it might be, well, that's a no-brainer. We know it's DNA. But no one knew it was DNA at the time when they did this experiment. The other thing that this experiment really gave us was this ability to radioactively label pieces for molecular biology. We do it now for lots and lots of things. So if you're going to insert a gene into a bacteria to try to get it to replicate that gene, you want that bacteria to glow that has the gene. And so you can label them. All kinds of situations where we're interested in this labeling technique. And it was really, they're not the first to use it, but it was the first to really use it in the sense of biological applications, especially to discover something like whether it's protein or DNA is the source of the gene. Okay, so from then we understood that DNA is the source of the gene, but in order to understand how DNA works, we needed to figure out what's the structure of DNA. And there was lots and lots of work that had already went into this. We knew prior to even Hershey-Chase work that DNA was comprised of nucleotides, and that nucleotides have three main parts. A sugar, if we're talking about DNA, a deoxyribose sugar, if we're talking about RNA, a ribose sugar, a phosphate group, and then some organic base, okay, one of the four that we know. Now, nucleotides differ with their bases, okay, and there are a couple, and we've talked about these before, there are a couple bases which we call large bases, or the purines, and they have a double carbon structure, double ring structure, and that's adenine and guanine. And then there are smaller bases, which we call pyrimidines, which have single ring structures, and that's cytosine and thymine. So if you remember, I talked about this quite a while ago, that adenine always binds to thymine and guanine always binds to cytosine, and that they form hydrogen bonds between each other. But one of the main reasons why DNA takes a double helix form and is uniform in shape is because of this bonding and the fact that you have a double ring always binding to a single ring. So it's the same width everywhere you look for a DNA molecule. Now this was noted and this was discovered by a man named Edwin Chargaff. And these are called Chargaff's rules. And Chargaff's rules suggest that DNA always binds in the same way. Adenine always binds to thymine, and cytosine always binds to guanine. And they bind in equal amounts. So if you have, 220,000 adenines in a sequence, then you will have 220,000 thymines in that same sequence. So they're binding at equal amounts. And it doesn't mean that mistakes don't happen and mutations don't happen. We'll talk about mutations a little bit later in a few lectures away. But Chargaff's rule really gave, especially Watson and Crick, it gave them this information that they needed to know in order to try to figure out the structure of DNA. Okay, so here you can see adenine binding to thymine, okay, double ring, single ring, guanine binding to cytosine, double ring, single ring. Now these molecules are really, really similar to each other. Okay? So there's a little bit of difference maybe on a double bonded oxygen, or whether or not they have a nitrogen coming off a carbon, but they're pretty similar to each other in regards to structure of the molecule, which makes them fairly useful in the case of building blocks. If you're building with the exact same or very close to the same Legos, you can make a pretty uniform model or uniform wall, and then the same thing is going on with DNA. Right. Now, I told you before that Rosalind Franklin, and I talked about her at the very beginning of the semester, and I put up a book that I think most people should read. Rosalind Franklin was integral and probably the biggest, the most integral part of discovering the structure of DNA. Rosalind Franklin actually took pictures, X-ray diffraction pictures, of DNA, showing that it was helical in shape. And for that matter, she also knew that it was probably a double helix. So before Watson and Crick, Rosalind Franklin took very good notes and was extremely hard worker. But a woman in even the 1950s, if you're really smart and you're really brass and you didn't want a guy to help you all the time or to be connected with all your time, you were thought of not being a very nice person and not a very nice word. And that's what a lot of people thought of Rosalind Franklin. But her work was amazing. It was excellent work. And it really was that Rosalind Franklin probably actually should get solo credit for discovering the structure of DNA, and we give her very little credit for it. If you look in her notes that have been published, she writes that DNA is most likely double helix well before it is published by Watson and Crick as being double helix. Okay. So Francis Crick and James Watson are the individuals that we give credit for discovering that DNA was double helix. Now again, I don't want to take too much credit away from both these individuals, but the amount of work that they actually put in to discovering the structure of DNA was nothing compared to Chargaff, and nothing compared to Rosalind Franklin, and for that matter, nothing compared to another individual named Maurice Wilkins. But Francis Crick and James Watson tied all the pieces together and actually deduced that it was double helix. At the exact same time, we've talked about another individual that's very famous, Linus Pauling, who came up with the protein structure, or the helix of a protein structure. He was the first to really describe protein structures. He also came up with a structure for DNA, and he said that it was a triple helix. So even though Franklin's information was out there, publications, Chargaff's information was out there, Even a brilliant mind like Linus Pauling said that it was triple helix. So I don't want to take too much away from Francis Crick and James Watson. They did discover that it was double helix, but most likely Rosalind Franklin already knew it was double helix. She was just collecting more data. She was very particular on publications. She didn't want anyone to come back and say, well, you're missing this piece, or you didn't think about this. So she definitely crossed all her T's and dotted all her I's. And in this case, she lost the race, if it was a race, to discover the double helix. Okay. Nonetheless, it was discovered that it was a double helix, that hydrogen bonds were the bonds that were between the bases, and that one purine would bind to one pyrimidine, okay, and that would keep a uniform, consistent thickness for the molecule, and that both strands, one strand would run from what we call 5' to 3', and the other strand is anti-parallel, so it runs the opposite direction. It runs from 5' to 3', and that's the formation of the double helix. Now, I'm not bashing Crick and Watson, because they did give Rosalind Franklin and Maurice Wilkins credit for the discovery of the double helix. Their publication came later, and they won the Nobel Prize later, and Rosalind Franklin had already died of cancer when Francis Crick and James Watson and Maurice Wilkins won the Nobel Prize for the discovery of the shape of DNA. So Rosalind Franklin was getting credit, not nearly as much credit as Francis Crick and James Watson, but she was getting credit for the structure of DNA. It's just that if you are dead, the Nobel Prize committee will not reward you or award you a Nobel Prize. So you cannot get a Nobel Prize if you're not living. So Francis Crick, James Watson, they're the individuals that are most noted for the shape of the double helix. Maurice Wilkins also won the Nobel Prize with them, okay? And that is kind of where we're going to end when it comes to DNA structure. Now we're going to talk about, well, how is it that it can break apart and reform and copy itself, and what happens when there are mutations. Okay, so next time we're going to talk about how DNA copies itself.